Okay, so I'm going to start off here. This is my presentation for the protein paper. Okay, start with the title. That's a good place to start. I went the what holds us together because that is pretty much literally what DNA ligase does. It holds DNA together. But let's start off with the first paragraph. The first paragraph is just general protein stuff. And I have a few pictures, primary structure, and it's just amino acids. I have a picture of an alpha helix. This is very nice, very good. Beta sheet. And that pretty much ends the whole generic protein stuff. Let's start in on actual DNA ligase stuff, my protein. Okay, so basically DNA ligase was discovered in labs in 1967 and 1968, two different labs that happened to find it pretty much within a year of each other. DNA ligase belongs to the ligase family, and it joins two molecules together. You can't really see any of the information on the camera, but oh well, it's a good thing I'm talking. And as I said at the beginning, DNA ligase joins DNA helixes together. It's also involved in DNA repair pathways and DNA replication. There are 3 steps in ligation. Basically, there are 3 steps. The first step is the activation of the DNA ligase through the formation of a covalent bond between an AMP intermediate. So basically what it's doing is the ATP goes into the molecule, and I'm going to assume that the little ladder here is, I don't know, covalent bond. That seems like what it is. Okay, so the little ladder is a covalent bond. Okay. The AMP intermediate is formed by adenylation, which is the addition of AMP. So there's adenylation right there. After the addition of the AMP, a phosphoamide bond forms between phosphate of AMP and the R of the amino group of a lysine. So, as we can see here, there's the addition of the AMP. And here's the molecule. So now it's attached to the R group of the lysine. The second step is the transfer of the nucleotide. Oh, whoops. It's actually nucleoside here. Stupid spell check. And that was the molecule that formed after the phosphoamide bond formed the 2 molecules. But anyway, the nucleoside is transferred to a phosphorylated DNA, and it forms a pyrophosphate link or bridge structure. And in the final step, the hydroxide of an adjacent DNA strand catalyzes the phosphorylated DNA end to release the AMP and covalently join the DNA strands. So basically, the AMP is just like a temporary thing. And now the DNA strands are joined. This process occurs in every organism that contains DNA. And that includes plants and eubacteria. The processes between the eukaryotic cells and the eubacteria cells, however, differ by the required cofactor and also what energy carrier the ligase is dependent upon. And when I say energy carriers, I mean like ATP and NAD. The cofactor is a non-protein compound that is required for the protein to function properly. For example, copper, zinc, or iron in ionic form. And like I said, the energy carriers are NAD plus and ATP. The eukaryotic organisms rely on ATP and have been separated into several groups. And those are ligase 1, ligase 3, ligase 4, and ligase 6. Ligase 6 is the plant-specific variant, and we'll get to that later. Ligase 1 and 4 are in all eukaryotes. Ligase 3 is observed only in vertebrates, and like I said, ligase 6 is plant-specific. In plants, the function of the DNA ligase 6 is rapid. It's supposed to be involved in rapid seed germination and seed longevity and quality. So, as I said before, the energy carrier that the ligase is dependent upon differs. And as such, the DNA ligase has differing structures of organization that corresponds to the energy carriers. The ATP-dependent structure is described as being similar to that of a crystal, and it has adjacent nucleotide binding domains and OB fold domains. OB folds are folds that are based on oligosaccharides and are 5-stranded beta barrels. And there's a picture of a beta barrel. It's pretty cool. And it binds ligands to the same face as used as an active site. So, in regards to the secondary structure, yeah, there are definitely alpha and beta helices and sheets within the ligase. I'd say that there are probably more. What's most notable is the beta barrel. Okay, and I have a picture of some of these ligases. Like, for example, there's the first ligase, ligase 1, that I talked about. This one is, I have no idea what that ligase is. I'm not even talking about it. But the one most notable is probably this ligase, the first ligase. Okay, so the first thing we need to note is right here, that's supposed to be like green. And right here is yellow. Okay, so the first step is the green. The green is the, we've got like a okay. The green is the adenylation process. That's the first domain. But it's also attached to a blue part. And the blue part, if I can find it up here. Yeah, there's a part right here. And that is the terminal amino group. It's basically where everything starts out. And there's the adenylation domain. Okay, and I think next is the red domain. The red domain is domain 2. That would be that one right here. And that's the OB folds that I talked about earlier. The third domain would be this orange one right here and this tiny itty bitty yellow one. The yellow one is the cofactor, the zinc or iron or copper that allows it to function properly. And the orange is, what is that? Okay, I have it described down here. The orange ribbon is the nucleoside. It's the described here as the H, lowercase h. I'm going to assume that means a nucleoside. There's a purple one right up here, and that would be the other terminal capping amino acid that ends our little protein. So that's the structure. Okay, and that structure was the one dependent upon ATP. I couldn't find any very good ones with the NAD plus dependent structures. Okay, I have a picture here which is the coding. You can't really see it. It's the coding for the ATP dependent ligase subunit. Yeah, so I was able to find that. There are several genetic diseases regarding DNA ligases, and most of them that I found are not fatal, but they're not good. In the case of DNA ligase 4, there's a mutation that occurs in the 4 gene, and it represents a major part of DNA repairing mechanisms. And it doesn't prevent the creation of the enzymes, but it reduces the ability of the DNA ligase enzyme to function. The symptoms are typically microcephaly, which is an undersized cranium due to either abnormal growth in the brain or from syndromes associated with chromosomal abnormalities. Unusual facial features, unusual growth, slow development skin abnormalities, and they're pancytopenic, which means the individual suffering from this has a reduction in red blood cells, white blood cells, and platelets. The individual who suffers from this mutation might also suffer from radiosensitivity and immunodeficiency. What the heck? Okay. And then I have my conclusion and my works cited. So that's been my presentation. I hope you enjoyed it.